I am Mr. Sandeep Dattatreya Kardule, M.Pharm Pharmaceutics, lecturer, Mula Rural Institute of Pharmacy, Sony, and I am giving presentation on extraction process part 2. Content of my presentation: percolation processes. In that, reserve percolation process, modified percolation process, continuous hot percolation process, then digestion, factors affecting choice of extraction process. A percolation process: the various percolation processes used for the extraction of drugs. First, simple percolation or petroleum process for tinctures. Second, percolation processes for concentrated preparations such as A. Reserve percolation process, B. Modified percolation process. Third, continuous hot percolation process. Or Soxhlet. Simple percolation process apparatus. Three types of percolators are used. First, conical percolators. The percolator is made of glass or metal, usually copper, which is tinned inside. It is conical in shape, having lower diameter not less than half of upper diameter. There are less chances of choking percolator in case the drug swells up, because a drug can slope against the wall of the percolator. Second, cylindrical percolator. The percolator is cylindrical in shape, that is, upper and lower diameters are same. When a higher concentration of alcohol or any other volatile solvent is used as menstruum, a cylindrical percolator is preferred. Third, steam jacketed percolator. When percolation is carried at higher temperature in order to increase the solvent action of the menstruum, the percolator is heated by a steam method. Simple percolation process is used for the preparation of tincture. There are three stages in the official method for the preparation of tincture: A. Imbibitions, B. Maceration, C. Percolation. A. Imbibition. The powdered drug is moistened with sufficient quantity of menstruum and allowed to stand for 4 hours in a closed vessel. Pack the moistened drug into percolators and add sufficient quantity of menstruum to saturate the material. When liquid starts coming out from outlet of percolators, the outlet is closed. Then the sufficient quantity of menstruum is added in order to leave a layer above the drug. Maceration: the moistened drug is left in contact with menstruum for 24 hours. During this period, the menstruum dissolves the active constituents of the drug and becomes almost saturated with it. C. Percolation: it consists of downward displacement of saturated solution formed in maceration and extraction of the remaining active constituents present in the drug by slow passage of the menstruum through the column of the drug. After collecting 3/4 of the required volume of the finished product, or when drug is completely exhausted, the mark is pressed. Mix the expressed liquid with percolate. Then add sufficient quantity of menstruum to produce the required volume and then filter. This is the diagram of packing of the percolator. On upper side, menstruum, below that washed sand, below that filter paper, then drug, below lower side glass full. Steps involved in maceration. During the period of 24 hours of maceration, the menstruum penetrates into the tissues of the drug and dissolves the active constituents. A short maceration before percolation enables the extraction of active constituent of the drug with considerably less quantity of the menstruum. After 24 hours of the maceration, open the outlet of the percolator and collect the percolate, which measures about 3/4 of the volume required for the finished product. The exhaustion of drug may be tested by doing the specified chemical test depending on the constituents present in the drug or absence of color in percolator. After exhaustion of the drug or after collecting the required quantity of the percolator, the mark is taken out from percolator. It is pressed to recover costly solvent. Then the expressed liquid is mixed with percolate, and then final volume is made by adding more of menstruum. The preparation is filtered in order to free it from cell debris or other material which interfere with clarity of the preparation. Example: tincture of belladonna. Second, compound tincture of cardamom. Third, strong tincture of ginger. Percolation processes for concentrated preparations. Percolation process for concentrated preparations are used for preparing liquid extract and solid extracts. The various processes used for preparing concentrated preparations are: A. Reserve percolation process. B. Modified percolation process. First, reserve percolation process. In this process, a part of the percolate, generally 3/4 volume of finished preparation, is reserved. Then the percolation process is continued till the drug is completely exhausted. Then the percolate is subjected to evaporation or distillation to convert it into a soft extract. This soft extract is dissolved in the reserve portion of the percolate, and then sufficient menstruum is added to produce required volume. Then modified percolation process. In percolation process of preparation of tincture, the drug percolate D by P ratio is about 1:4. The D by P ratio is reduced to 1:3 by modifying percolation process, and hence there is a lot of saving in heat, time and menstruum. Percolation is a displacement process. The strong solution of active constituent of drug formed during maceration is displaced by a fresh menstruum when percolation process is started. It is proved that stationary menstruum remaining in contact with drug dissolves more matter than flowing menstruum. Hence, more menstruum is required to exhaust the drug when simple percolation process is used. But if continuous percolation stage has suitable breaks by short maceration stages, the D by P ratio can be reduced to 1:3. Then continuous hot percolation process or Soxhlation or Soxhlet extraction. When active constituent of the drugs are not freely soluble in the solvent or difficult to be displaced from the cells of the drug, then it becomes necessary to extract the crude by action of hot menstruum for considerable length of time. The fixed oil from seeds and alkaloids from the drugs are extracted by continuous hot percolation process using benzene, chloroform, petroleum ether, etc. Apparatus. The apparatus used for continuous hot percolation process is Soxhlet apparatus, which consists of three parts: A. Flask containing the boiling solvent. B. Soxhlet extractor, in which drug to be extracted is packed. It has side tube which carries vapors of the solvent from the flask to the condenser, and siphon tube, which siphon over the extract from Soxhlet extractor to the flask. C. A condenser in which vapors of the solvent are condensed again into solvent. This is the diagram of Soxhlet apparatus. Upper side, cooling water out, below that cooling water in. Then middle there is a thimble, distillation path it is shown. On extremely lower side, steel pot is there. In that stirrer, siphon exit, solid drug, siphon top, expansion, then upper side condenser. Below heat is provided by heating mantle. Procedure: the drug to be extracted is packed in a paper cylinder made from a filter paper, and it is placed in the body of Soxhlet extractor. The solvent is placed in the flask and apparatus is then fitted. When solvent is boiled on heating the flask, it gets converted into vapors. These vapors enter into condenser, trough the side tube, and get condensed into hot liquid which falls on the column of the drug. When extractor gets filled with the solvent, the level of the siphon tube also rises up to its top. The solvent containing API in the siphon tube siphon over and run into the flask, thus emptying the body of extractor. This altering of filling and emptying the body of extractor goes on continuously. This process is repeated until drug is exhausted. The process is repeated about 15 times for complete exhaustion of the drug. Limitation of continuous hot percolation process. Physical character of the drug. The physical character of the drug is such that it would block the Soxhlet apparatus in case it is used for its extraction by this method. For example, opium, gum, resin, orange peel, etc. Solvent. Only pure solvent or constant boiling mixture can be used for this process. Then chemical constituent of the drug. The process is unsuitable for drugs having thermolabile active constituent such as enzymes, alkaloids, anthraquinone derivatives, etc. Digestion. In digestion process, the drug is extracted by heating at a particular pressure. This will increase the penetration power of the menstruum so that there is complete extraction of the drug. Precautions should be taken so that increased temperature may not harm the active constituents of the drug. The apparatus known as digester is used for extraction of the drug by this method. It is a vessel made up of metal. The whole of the drug along with the menstruum is placed in the body of the digester. Place the cover over it and bolt it with the help of nuts. The drug is treated with menstruum for a definite period under a specified condition of temperature and pressure. Factors affecting choice of extraction process. First, character of the drug. Second, therapeutic value of the drug. Third, cost of the drug. Fourth, stability of drug. Fifth, solvent. Sixth, concentration of product. Lastly, thank you.